A river flows from a mountain spring to a delta at the sea. Along the way it branches. One arm flows east, another west. You drop a net at a random point and catch 100 fish. Some are near the source, some near the mouth, some at the branching point. You caught them all at the same moment, but they're at different stages of the journey. Differentiation works the same way. Cells captured at one time point are at different stages of a biological process. The pseudotime reconstructs the order. Single CellRNA-seq captures a snapshot. Every cell is fixed at one moment. But differentiation from stem cell to neuron, from progenitor to mature T cell, is a continuous process spanning days or weeks. The cells in your dataset aren't synchronized. Some are just starting. Some are halfway. Some are nearly finished. The expression profiles of early cells differ from late cells in predictable ways. Pseudotime analysis exploits these differences to reconstruct the temporal order from a single static snapshot. Pseudotime is not clock time. It's biological progress along a trajectory. Cell A has a pseudotime of 0.2. It's 20% of the way through differentiation. Cell B has 0.8. 80%. The ordering reflects transcriptional similarity to the starting state. Cells with expression profiles close to the stem state get low pseudotime. Cells with expression profiles close to the mature state get high pseudotime. The key insight: you can infer temporal order from transcriptional similarity without longitudinal sampling. Monocle and Slingshot fit a principal graph, a tree or smooth curve, through the cells in PCA or UMAP space. The graph defines a trajectory structure, a single lineage, a bifurcation, or a complex tree with multiple branches. Each cell is projected onto the nearest point on the graph, and the distance along the graph from the root gives the pseudotime value. Branch points in the graph correspond to cell fate decisions, where the river forks. Monocle 3 uses a principal graph learning algorithm called SimplePete. Slingshot fits principal curves separately for each lineage. Diffusion Pseudotime takes a different approach. Build a kin graph connecting similar cells. Compute the transition matrix, the probability of moving from each cell to its neighbors. This is a Markov chain from the board game video. The diffusion distance from a designated root cell defines pseudotime. Cells that are many diffusion steps from the root are further along the trajectory. Diffusion pseudotime is robust to noise because it averages over many possible paths through the graph. It naturally handles branching. Different branches have different diffusion distances. Every pseudotime method needs a starting point. Where is the source of the river? If you know biology, the cluster expressing known stem cell markers is the root. If you don't, or in a velocity, can infer directionality. Cells with positive velocity for differentiation genes are moving forward. Alternatively, compute a stemness score. Cells with high expression of pluripotency markers and low expression of lineage markers score highest. The root choice matters. Reversing it reverses the entire pseudotime ordering. The snapshot problem: one time point, many stages. Pseudotime ordering cells by biological progress. Monocle and Slingshot, principal graphs in reduced space. Diffusion pseudotime, Markov transitions from a root cell. Root selection, biology, velocity, or stemness. Next, which direction are the cells actually swimming?